everyone. Good morning, good afternoon, good evening. Oh, I think we made it to day 3. I hope you're awake, comfortable, happy, enjoying yourself wherever you are in the world, and craving for more MEEG lectures. With a few glitches, hiccups, this gardening adventure seems to be working, don't you think? Okay, let me take a short moment here to introduce you the Cutting EEG crew behind the scenes. Let me start by introducing you to Christelle Zielinski, aka behind the scenes. That is an engineer at the Laboratoire Parole et Langage in Aix-en-Provence. She's taking care of broadcast connection troubleshooting, covers the questions sent by the gardens with Anaïs. And this is Anaïs, your Laboratoire Parole et Langage in Aix-en-Provence. She's a researcher at the IPNP in Paris and Fanto Besançon. She's taking care of passing the question to the chair, displaying them on the screen, monitoring the constant streams of messages and signals. And this is Clément François, aka Black Pepper. Clément is a researcher at the LPL in Aix-en-Provence. He is taking care of scientific and provocation advisor. And the apéro, most important job of the day. And let me take you to François Tadel, aka Sage. François is an independent researcher engineer based in Grenoble. He's taking care of general brainstorming and mind opening. Let me introduce you Alexandra Corneli, aka Donkey Bean. Alexandra is a research engineer here in Lyon at CERNL and taking care of inventing a new kind of scientific meeting here in Lyon, providing a perfect environment for our staff team. Do I need to introduce Maximilien Chaumont? Maximilien, aka Fenner, is a research engineer at the Paris Brains Institute, founder and co-president of Cutting EEG. He's taking care of administrating the stage and, of course, the global coordination. Let me bring you to Hervé. Hervé Guenet, local staff here in Lyon. Let me take the camera here and I'm showing you the behind the camera. This is Adrien Schramm. Adrien is our favorite independent organizer. And he's fancy. Thank you, Adrien. All right. Now, I am, I think, the last seed in the pot. My name is Anne-Sophie Duberry, aka Cardamon. I am a research engineer at Laboratoire de Neurosciences Cognitives in Marseille and co-president of Cutting EEG. I'm handling technical aspects and montaging the videos. I'm helping to select the questions for the speakers. Now, if you take all of these plants together, you mix them the good way, you can end up with a good pastis EEG. Enjoy the day, have fun, and stay tuned.
Hi, it's a wonderful video. So thanks for joining us today. We have the Reproducible Processing Pipeline and Multiverse Analysis session at Cutting EEG Conference today. I'm Yu Fang, who's postdoc at Fire University Berlin, and today I'm the chair going to navigate this session with you. And this is fantastic to see all the people here today who would like to dive into this intricacy of EEG analytical pipeline and a multiverse analysis. So today we have a treat with the 3 brilliant speakers, each bring a unique perspective on the EEG many pipeline projects, ERP multi-analysis, and to Artemis projects. So kick off the session, we firstly welcome Elena, who had head of EEG many pipeline projects and analysis EEG data. So Elena, over to you. And then now she's going to share the screen with everyone. And then don't forget to ask the questions. If you see the chat next to it, there's a question mark. Please ask your question there. Okay, I hope you guys ready. And Elena, the floor is yours.
Thanks, Yufang. I think the first thing we test is if you can hear me well and if you can see the screen well.
Yes, we do.
Yeah, all is fine? Great. If something happens, just interrupt me and we can go on from there. So thanks a lot for the invitation, and thanks, Yufang, for a short and nice introduction. I'm very excited to give a talk about the project I feel so strongly about, which is called EEG Many Pipelines. I believe many of the people that are listening here, since this is also a Cutting EEG organized event, might have heard about the project, but I'm very happy to introduce the whole philosophy of it as well. In a big picture, it's a multi-analyst project that is focusing on the variability of the pipelines in EEG preprocessing domain and how it's linked to the results. I'm not doing this work alone, so together with me, there is a group of wonderful research scientists working from all over the world, which we call steering committee, and it's just a short slide to represent them as well. So to begin with, reproducibility should be the defining feature of science. However, you're most likely familiar with the concept that there is a reproducibility crisis in psychology and cognitive neurosciences. Perhaps you also saw this publication that was released in 2015 from the Open Science Collaboration, which I think is illustrating it quite nicely, where the authors tried to replicate 100 studies in psychology. And this is what they found. On the x-axis, you can see the original effect size of the original publication, and on the y-axis, you can see the replication effect size. In the ideal case scenario, all the effect sizes should fall onto this diagonal line and be in the green bubble, which would represent a significant finding. However, we did see that we do see that it's not always the case, and not all studies nicely map on it. Some of which could also not be replicated. So there are many reasons what could be the reason behind the reproducibility crisis in the field. And one of them is variability, potential variability in analytical choices. That is the one we addressed further in this presentation as well. So there have been attempts to investigate this, and this was a kickoff project in 2020, where a group of scientists, they sent an fMRI data set to 70 independent laboratories in the world, and they asked them to answer the same set of 9 hypotheses. The result was striking because they report that analytical flexibility in the way that scientists analyze the data, preprocessed it, had substantial effects on the scientific conclusions. I think this paper, when it was released, it received a lot of attention from the scientific community. And it also resulted in the fact that we also asked this question in our neuroimaging domain. So how about the EEG data analysis practices? Are they also that variable? And what is the effect of on the result that we get? Are they comparable to one another? With this idea in mind, our steering committee member and the founding father of EEG Many Pipelines, Tom Marshall, posted a question in Twitter in 2020, just after the publication was released. He asked people, basically the research scientists of EEG and MEG research, whether they believe if we did exactly the same study, if we sent the same EEG data set to 70 laboratories in the world and asked to analyze them in the way this data in the best way they know how, to answer a simple P300 related question, whether our results would be more or less consistent. What I like is that he got 601 votes. So it definitely increased it showed the interest from the community. And most of the respondents did say that they believe our results in EG would be more consistent. This was the launch of the project, and it started completely voluntarily. So a group of researchers joined together, they met online. It was, as you can also see, the pandemic time. So also I think all the conditions were there to set such a project of a community driven by the community in this research field. So we teamed up and sent the data set. We prepared the data set and asked teams to sign up to the project. And I'm happy to say that we had more than 300 laboratories in the world that signed up to the project initially and that received the data set. After the data analysis phase, we were happy to receive a complete results from 168 teams that successfully finished the data analysis phase, which was a substantial effort from those parts as well. And this is a nice map showing the distribution of these teams, where most of them were coming. So most of the teams you can see that come from Europe, but we have a nice coverage over all the globe. So it also includes the US, Asia, as well as Australia. This is now also, so the idea of the project, as well as the description of our analyst teams and the whole setup is described in the preprint of our position paper that is now on Meta Archive. It's currently under revision, and we hope it will be soon published in a peer-reviewed journal as well. I won't go a lot into detail about the position paper for the sake of time, but if you would like to know a little bit more about our sample, what it consisted of, and what are the characteristics that define our teams that are important for multi-analyst projects, you can have a look into this paper. So just to shortly sum up how our experimental design looked like, we sent these 106, more than 300 teams behavioral data that consisted of a simple memory task, also containing human-made and natural stimuli that were presented to participants. We also recorded EG data, which was multisensory EG of 33 participants that was sent to the analyst teams. And we asked 8 hypotheses. Now, it's important to mention that these 8 hypotheses varied in the level of specificity. The first ones were really specific. This is an example of one. So we basically said in the first hypothesis that there is an effect of scene category on the amplitude of N1 component. And the less specific one was, for example, hypothesis 3b, which stated that there are effects of successful recognition of all images, so those images that were presented not for the first time, on spectral power at any frequencies, any channels, and any time. You can see that it's less specific. We're not predefining what kind of a frequency range to look at. So all of this set of data were sent to the laboratories, and I think we gave them roughly 8 or 9 months of time to analyze it, and we received data back. What kind of data did we receive back? So we asked for pre-processed time series data. We also asked for the scripts that were used to analyze the data. We asked to submit result forms that would detail the results that the analyst teams got after they analyzed the data. And we also asked to fill in every analysis questionnaire. And those were the questions we came up with the steering committee members. And in total, there were around 600 questions that we thought were important to detail the pre-processing pipelines that were used. In total, maybe it's an interesting fact that we received around 5 terabytes of data. The way I see the project, I think it contains 2 main goals. So the first one is to observe variability in the analysis pipelines and the results, because up to this date, although we almost next year will mention 100 years of the discovery of EG, we don't know what are really the analysis pipelines in the world, because codes are often not shared, and it sometimes really feels like laboratories are working independently from one to another. The second goal is to link the 2. So we want to link these analysis choices to the results. And now we're in the middle of the project. And for this reason, I think we now have quite a good understanding of the first goal of the EGMA pipelines, and I will focus on it a little bit more today. So how variable are the pipelines? Maybe to start with, we have two sources of data where we could look at to answer this question. The first one is the analysis questionnaire that we sent to the analyst teams, and that represents the self-report of the teams. So basically, they answered the questions we asked to specify what were the parameters that they used, whether they did this step or not, and they could answer it. The second source of data that we could get, that we could answer this question, comes from the scripts. And well, we believe that it is important to compare the two for several reasons. And I think this was one of the biggest tasks we had over the last year, was to look into the scripts and to try fill in the missing reports and several types of posts that we could find in the self-reports. We came up with a semi-automatic script check procedure that I can't go into detail too much into, but if you're interested, you can always contact me. And we checked codes of 168 teams. So just to give a bit of numbers, those were 1,200 files, roughly more than 1,200 files. Each of them contained codes to analyze and pre-process the data. On average, per code, we saw that most teams had 1,700 lines to process the data to answer these hypotheses. And by using this approach, we were able to correct misses that we found in the reports. So some parts that we thought are substantially important to answer our questions, and some small inconsistencies. So typos, maybe scales, and everything that we had a question for, we looked at the script check. And this is a short glimpse into what kind of variability do we see. These are just only 5 of the parameters that we already looked at, but there are much more. So with the first glimpse, you can see that some of the choices are highly variable and others less so. What I find quite interesting is to also firstly look in the software that we have. So our 3 main softwares that our analyst teams used were EEG-Lab, MiniPython, and FieldTrip. I think it's not very surprising, but it's also nice to see here. And most of our analyst teams had quite a good knowledge of these softwares that they use, because this is also represented here, denoting that most of the teams had from 2 to 10 years of experience using the software, so they're really experts using it. Most of the people will reference their data. Most likely choice is the average reference. They also often tend to downsample. However, it's not always the case. The original sampling rate was 512 hertz. The most common way to downsample is to 265. So almost all teams, low pass and high pass, filter their data. And again, you could, with a short glimpse, you could see that low-pass filters are much more variable, so there are much more options that have been used there. So pipelines seem to be variable. Another interesting metric to look into it is the order of steps. So we also asked analyst teams to report in which order they perform these 15 steps that we specified. So these steps are on the x-axis. They start from downsampling to filtering, and they end with baseline correction. On the y-axis, you have the order of steps. So basically what this graph represents is that the bigger the circle, the more teams perform this step. The same information is also held in the color, so also the darker the circle, it also represents that most teams match in the order of steps for this particular preprocessing option. For example, here you could see that most teams high-pass filter their data as one of the first steps. Same goes for low-pass filtering. And as we go further down the line, you see that it's almost diagonally increasing. Some of the steps also are more variable than others. For example, downsampling really happens almost at different stages of the preprocessing pipelines. We then thought whether it would be interesting to see if this variability would be reflected differently given the different softwares that people are using. And this is what we looked into as well. So this is now divided by the software that teams are using and are the team sizes that we had. Again, the biggest is EEG-Lab if we combine with all the plugins that were used as well. And here we see some also quite interesting variability that exists in our data that also might be specific to the software that our analyst teams were using. So just by eyeballing it, maybe a few things that we noticed as well was, for example, downsampling. Downsampling seems to happen quite early on for the EG lab team. So as one, if teams performed it, it was performed as one of the first steps, which is not entirely true for the teams that used MNUPython, that where downsampling appears as rather in the middle of the pipeline. The same true is for field trip. It was also interesting to observe that epoching is another thing that also seems to be different among the teams using different software tools, which while for teams that use field trip, it looks like epoching often happens as rather early in the pipelines as one of the first steps, which is not true for teams that use MNUPython or EG Lab, which epoching then rather happens in the middle of the pipeline and almost never at the very beginning. The same is true for Brain Vision Analyzer, although we have fewer teams using it, but we thought it's also very interesting to look into this because conceptually it's quite a different way of processing your data. And here it also looks like more teams perhaps match in their analysis choices because we have bigger circles basically denoting this. Now, how variable are results? So we plotted whether the hypothesis was confirmed or not. And here I think variability is substantial. However, it really depends on how specific the hypothesis was. So again, I just want to remind that the first hypothesis was rather specific and the last one was very unspecific. And it's not surprising that many teams agree on the first two hypotheses. However, not all of them, but they, for example, find significant effects confirming these hypotheses. And this is less of the case for A or for B, where the formulation of the hypothesis is really not that specific and allowed for exploration. I wanted to show you also how these results look like. Again, I think we should take it with a grain of salt because analysis choices are highly variable and they are hard to compare one to another. But if we step back and we just want to look how do the results look like in general, disregarding whether analyst teams used cluster statistics, all of the channels, or predefined one or two channels, just to step back and to see if I was curious about this effect that the first hypothesis was meant to test, what were the results I would find in the literature or that our analyst teams, in the way that our analyst teams were reporting it. So in the context of EG-mini pipelines, for the first hypothesis that I presented a little bit before is that there is an effect of scene category on N1 amplitude. We saw that 135 teams confirmed this hypothesis, so we found significant effects. From those teams that found this significant effect, this is a topography which channels were included in the significant effect. So if the channel would be gray, that would mean that not a single team found a significant effect in this channel. However, we saw no gray channels, meaning that at least one team found the effect in at least one channel. So all of them were covered. The warmer colors represent that this is a convergence between the teams. So that's where most of the teams find the effect. And here we can see that it's mostly in parietal occipital areas and central areas as well. Now, the time window that they analyzed, again, was quite broad, variable in the sense. Here, if you can see, the dark blue color represents the time window that was analyzed, and the light green color represents that this is a significant window that was found. So most of the teams actually did analyze the time window around 100 milliseconds post-stimulus presentation and found the effect there. However, it's still variable, so it can be broader or smaller depending. What it does not always, because our initial thought was that the bigger time window, the bigger the significant, the longer the significant effect. It also seems like it's not always the case, at least from this graph. Now, we also looked at a hypothesis that was less specific. So 3B, the one, again, I can surely remind you that there's a time frequency hypothesis where we were very vague and specific. on the location or the frequencies that the teams should look at. And here we did see that it's not that clear whether the hypothesis was confirmed or rejected. So it's almost 50-50, depending on the analysis choice. If we would look at the topography again, it's mostly covered by warmer colors, meaning that more electrodes are included in the significant findings and more teams find them. But it's again widely distributed all over the cortex. The time windows are broader and more variable. So also, I think representing that data exploration is more likely to happen with a vaguely specified hypothesis. And this is again expected, but it was interesting for us to look how analysts are making these choices and what are they based on. The time windows again were varying highly, so it could vary from a second until stimulus till 2 seconds past it. Since that was the time frequency hypothesis, we also looked which time frequency ranges did the analyst teams look at, and this is the top figure right here. So that's the analyzed frequency range. We can see that frequencies from 0 to 200 hertz were analyzed, and below you can see the significant frequency range that we find significant. And they also reported that they found significant effects between 0 and 150 hertz for at least one team. However, most of the teams did look in the frequency ranges between 4 and 30 hertz, and most of the significant findings are also found around that region, but also in those lower frequency ranges. Okay, so how can EG-many pipelines help solve reproducibility crises? So our big goal is really not to find what's wrong or not. We really want to only identify commonly used steps. So we really want to map the pipelines that exist in the world and how they're used. And we also want to understand whether there are steps in the pipelines that could lead to certain results. So what should we do about it? So this is really the main purpose of the EG-many pipelines in the way that I see it. Based on these results, we want to provide guidelines and practical recommendations, meaning that if we were to discover that certain steps are more likely to introduce variability in the results, we do want to provide guidelines, how to what to do about them and what to perhaps avoid and be a little more cautious about. We also want to open a philosopher discussion for the future of reproducible science, because we think that's the way further. And by the end of the project, which is at the beginning of 2025, we will share this rich and open data repository that will include all of the data and all of the codes that we have used and we received with the rest of the community, so that more research can be carried on. So with that, I want to thank you for listening, and I want to thank our steering committee that is highly included in the project. And this is a picture from our first in-person meeting in Stockholm. We look all very happy about that we could finally meet after 2 years since launching the project. Thanks.
Okay, thanks, Elena, for the wonderful talks. So now, because of time, we now would like to take 7 minutes break to allow local and discussion. And then after 7 minutes, we will turn on back the camera for the questions for Elena. Thank you. So now we will have the 7 minutes for discussion. Yes. Okay, so the first question to Elena, the question is how variable are effects when teams use same time window, frequency range, etc., which means how many variation is in pre-processing versus statistical analysis choices.
We didn't look into the variation of the statistical analysis choices. We did constrain this observation to pre-processing steps because, it had to be. Then we will look into it further on. If you would ask me for my gut feeling for what, because since I was looking into the data, I think the variability in the inferential part of the analysis choices are going to be higher than in the pre-processing part, because it was really I think more things come into interplay there. So there are, and more degrees of freedom are also there. So it's not only the analysis, like statistical model choice, but also how, what is the threshold that people are choosing, whether they correct for multiple comparisons or not? What is the judgment inside, taken overall? There are many things that are sensitive to this kind of variability in the result.
Mm-hmm. Yeah. So another question, like, do you look at the influence of the software on hypothesis rejection or confirmation, which may do people use different like toolbox get different results?
We will look into this. We do believe the software might be one of the most important players in the variability of the results that we see. This is the next step. So we now move into the analysis phase of the project. As I'm talking to it now, we finished the first part. The second part after basically starts in October, where we are addressing exactly these questions. There was also a great question here before, if we know which software would produce more significant results. We do not know, but we will definitely include it into our models, as we believe this might be one of the very important players. However, I would not look into it individually, without including other parameters into the models, because it might still produce biased results. I think once we build models, we have to include all of the processing steps that we believe that might affect the result, and then see how it affects when other parameters are kept constant.
Another question is, do certain steps, like analytical steps, produce the highest bias?
If certain, can you repeat the question?
Is any specific like analytical steps, yes, produce the highest bias? Perhaps from your talk, we can't really tell, right? Because we haven't really looked into the steps, which one will produce the result, highest bias.
Yeah. This question comes a lot, and we cannot yet tell which one that produces the highest bias, because this will be at the end of the project. I thought it was highly important for us to map this variability in the pipelines as well as we can before we connect it to the variability in the result.
Mm-hmm. True. And this one is interesting because we talk about the script and the line of the code. So how often do people use built-in functions versus the program and use their homemade, like the script? Do you know that?
Yeah, thanks for the question. I also, it's the most recent task we finished in EGMA pipelines, and I think it's really great. I think it will also help many scientists in future who want to go through unfamiliar codes or huge amounts of it. I think most people that use toolboxes use inbuilt pipelines, inbuilt functions, and there is a lot of overlap between them within specific software. We're looking into it as well. It was also quite interesting to see this completely automatized pre-processing functions that have also been used. I think we'll include them into the models as well. I think we had around, not too many, but maybe 5-10 teams that reported that they completely use their own custom written scripts, but the rest mostly are inbuilt functions. However, it's also true that there were also quite some deviations between the toolboxes. For example, EGLab and Fieldtrip are often used interchangeably, so certain things are done in EGLab and other things in Fieldtrip. That was quite commonly the case, but even then, many functions are actually built for that software.
Mm-hmm. So, and the next one, because you're talking about the hypothesis, right? You show some questions. So some audience wonder, were the group's analysis team that involved in this project familiar with the topic of the study?
That's a good question. We did ask for a certain level of knowledge. So the way we addressed the question, We selected the analyst teams based on our inclusion criteria, and one of them were that the teams should have at least one publication in the field of in EEG in a peer-reviewed journal. In this way, we kind of assured that the teams that are participating in the project are familiar with EEG analysis practices and are doing them it themselves. So this was our controlling criteria. If they exactly, I mean, the level of familiarity with ERP or time frequency specifically has not been measured.
Thank you. The last questions, because you show the brain vision and some field trip and each lab. So audience wondered, the GUI solver things underrepresented in the study. Do you think that it could be have biased the results?
Sorry, I just read the question shortly. Yes, if they underrepresent in the study, that's a good point. There might be, for sure, we have smaller numbers of these teams, but I do think that since we have quite a big sample, it does represent a general trend in the field that we're dealing with. So I think it's true. They might underrepresent the teams that are using these software tools. But generally, what we actually also did, we compared where most of the PubMed publications in the field are coming from, based on the country location, and it mapped really nicely with the distribution that we have in EEG pipelines. This is a little bit different from, I think, the question that originally was asked, but I still do believe that it might underrepresent, but it gives a good understanding on what are the practices and what are the toolboxes that are most commonly used in this research. So it answers at least those parts of the questions.
Thank you, Elena. We have more questions, but because the time restrictions, so we're gonna start from now. And then we do have more questions, but yes, please also follow the project and over the social media or write an email or something. Thank you so much, Elena. And now we're going to navigate to another topic now. Up next, we're gonna have Peter Claessens. So he's going to take us on a journey through the multiverse of ERP research. He's going to show us the importance of non-just data process. Data, exciting to hear you and have you here. And please come on stage. Yes, your slide's ready. So here you go, got 20 minutes.
So I'm really excited to be able to talk to you and to get to tell you a little bit about some of the work we've been doing. But I wanted to start off with a fun fact about me, and that's that I love to cook and bake. So one of the reasons I love to cook is that you can kind of do what you want. You know, I really like garlic, so if it calls for a few cloves, I add 6 or 7. Same thing with cheese. The more cheese, the better. But baking is a bit more troublesome because it requires precision. So one of my favorite things to bake is chocolate chip cookies, but they're actually pretty easy to screw up. If you replace baking soda with baking powder, you get this flat, crusty cookie. If you don't refrigerate your dough before baking, you end up with a firm, crumbly cookie. It doesn't have that satisfying outside crunch with that delicious, gooey, fulfilling middle. And if you melt the butter, instead of letting it soften to room temperature, you end up with a greasy cookie that spreads out too much during baking. If you want the perfect cookie, you have to follow all the instructions precisely. You can't take shortcuts. But if you follow the recipe closely, as best as you can, you'll be rewarded with this beautiful, delicious cookie. So as you can see, baking it's not like cooking. You cannot just swap out ingredients, change proportions, alter the steps. And this is why they call baking a science. And when you want to help someone else produce the perfect cookie, you provide them with a detailed recipe. So when we want to help other scientists produce our cookies or our beautiful waveforms or replicate our findings, we write detailed method sections that communicate all the important steps of a given study. But it takes many, many steps to make files and files of these wavy lines like you see there on the screen into an interpretable waveform that you can do something with, like at the bottom right. And some of these replication difficulties that were just talked about by Elena, some of these replication difficulties in psychological science, they've really focused attention on the methodological ambiguity that contributes to these replication failures. Because if you don't report your recipe in its entirety, you might not be conveying important experimental details or pieces of your recipe. And it's quite difficult to reproduce or replicate a study if what was done in the first place isn't clear. Such ambiguity or missing information can elevate false positive rates, because in the face of this undisclosed flexibility, there are a lot of researchers' degrees of freedom that can be capitalized on, especially in neuroscience studies, because collecting and analyzing neuroimaging data is computationally intensive and requires many methodological choices. This missing information leads to a lot of wasted resources. So in biomedical sciences, it's estimated that billions of US dollars per year are lost due to misreporting of information and inadequate reporting of methods, because incomplete recipes yield to disgusting cookies that just waste ingredients. In appreciation of these issues, publication guidelines for ERP studies have been published. So this references one that was published by a committee convened by the Society for Psychophysiological Research, and there are many such reporting guidelines papers for studies of ERPs. But these papers help facilitate transparency and improve replicability by identifying the key parameters that should be reported in every study. And adhering to these guidelines ensures that these key details are reported and communicated to the scientific community. But I wanted to know a few years ago, or we wanted to know, how well are these guidelines actually followed? So we scraped articles of ERP studies across 5 journals that publish a lot of ERP studies. And we chose 5 articles per journal per year, which led us to code 150 studies for this article guidelines paper that was published by Psychophysiology. And here are the kind of things we were looking for. So participant information. Did people report the age of the participants, sex, years of education? EEG recording information, so information about the sensor make and model or online filtering, the stimulus and timing. What was the intertrial interval or the interstimulus interval? If you're using something like emotional images from the IAPS data set, which images did you use? That's the kind of information that's needed to confidently replicate a study. What were the pre-processing steps? Did you re-reference or interpolate bad electrodes, and how were those identified? How did you measure your ERPs? Did you use a peak amplitude procedure, maybe a time window mean amplitude? Which sensors? What statistics did you use? Did you check for violations? Did you correct for multiple comparisons? Information about PCA and ICA. So if the parameter wasn't reported in any given study, then it didn't need to be coded for that article. So for example, if an article didn't use PCA or ICA, it didn't count against it. But I think here are some illustrative examples of the kind of things we encountered in this project. EEG data were re-referenced. So that's pretty unclear without knowing how the data were re-referenced, such as algebraically to average mastoids. The EEG data were filtered, and it gives the cutoffs. So it's not enough information to confidently replicate this. Were these half amplitude or half power cutoffs? What type of filter was used? What was the roll-off? ERPs were baseline adjusted. When? And then if you're talking about scoring, how are you scoring? What's your approach? Which sensors are you caring about? So we coded these 150 studies across these 5 journals for these key parameters. And only 63% of these key parameters were reported on average, which is pretty bad. The range is from 39% to 82%. So not a single study reported all of this necessary information advocated by this guidelines paper. And there was no difference in performance across the journals. Now I'm going to show you the results by category. So the percent of parameters reported will be shown on the y-axis, and then each category will be shown on the x-axis. And as you can see, there's quite a bit of variability. You know, we're reporting more information about participant information and stimulus and timing, and comparatively less about ERP measurement and ICA, which seems really important. But the take-home message is that there's a widespread of omission of about a third of the required reporting parameters, which presents a substantial barrier to any replication efforts, as well as efforts to evaluate the quality of research. It's unclear how to judge a study and judge its experimental manipulation or data quality or data collection and processing if what was done is unclear. I don't know if this is comforting, but we are in good company. This is not unique to studies of ERPs. There's a fairly routine failure to not report all these key methodological parameters in fields of neuroscience and other biomedical sciences. And this could be due to an underappreciation of whether these data processing parameters actually matter for a given study. So maybe the reason people don't report this information because they believe it doesn't make a difference. Something that I think the many pipelines project has certainly helped us understand. We kind of took a different approach. So we took a multiverse analysis approach to identify kind of the key data processing stages in a study of the error-related negativity. Because it takes a lot to get files of these wavy lines into something smooth and interpretable like we see on the right. So we typically filter our data in some way. We'll use an eye movement correction procedure. So maybe you use a regression-based approach or an ICA approach. We often re-reference offline. We baseline adjust using some time window. The sensors used for scoring. So we're using a single sensor, a region of interest approach, which sensors, and different kind of measurement procedures, such as a peak amplitude, time window mean amplitude, absolute peak, etc. So we wanted to know what happens if we look at the impact of all of these steps simultaneously on an ERP, because any given study does different things for each of these steps. There's no clear consensus on how data should be analyzed or what is the optimal way to look at data. So we were interested in examining the error-related negativity. I want to briefly mention it's a negative deflection in the scalp-recorded ERP. It's associated with early error detection. So it's relevant for performance monitoring aspects of cognitive control and represents a manifestation of anterior cingulate cortex activity. Now I want to show you what the ERN looks like. So amplitude is on the y-axis, negative is plotted down, and time is on the x-axis. So we have error trials in red, correct trials in blue. The arrow there is pointing to the deflection that is ERN. So you can see the ERN is much larger or more negative for error responses than correct responses. And it's pretty clear to distinguish in this waveform. It also has this nice frontal central scalp distribution. But these waveforms represent just a single instantiation of a multitude of data processing pipelines that we could have used to analyze ERN for this figure. Just like there are hundreds of recipes to bake chocolate chip cookies, there are hundreds of recipes, if not many more, to look at ERPs and score them and analyze them. So the question is, how do we optimize data processing? How do we decide what is an optimal approach for a given purpose? Because it's likely going to vary depending on what your interest is, whether you're interested in something like data quality or experimental effects or internal consistency or group differences. And one way we could go about doing this is to look at data quality. So I want to show what data goes into data quality. So I'm showing 6 real error trials from a real participant. Amplitude on the y-axis, that shaded region is the window we use for the time window mean amplitude of ERN to score. And as you can see there, there's quite a bit of variability in those 6 trials. We can plot the single trial estimates for the single participant who made 69 errors. And we can focus on that between trial variability as an estimate of data quality. So if these ERN scores are very similar to each other, then we assume that would mean high data quality. But if they're highly variable, then that would indicate poor data quality. Because we assume that on any given trial, there's this true ERN signal and then some error that's hopefully random, which is why we're able to average many trials together to get a smooth waveform for a participant like this. So here I'm showing this single subject average for correct and error trial, ERN. Another way we could optimize a data processing pipeline is trying to maximize these within-person differences, so the between-condition difference between correct and error trials. So I could try to optimize a data processing pipeline for either data quality or experimental effects. But this really goes to show, I think, how characterizing data for a participant with one estimate can be really hard. A lot goes into this single ERN score of negative 1.9 for this person, and a lot of information is lost along the way. So we considered a data processing multiverse. and simultaneously looked at almost 3,500 ERN scores per person in an effort to maximize data quality and maximize experimental effects. So we wanted to know which data processing parameters led to the lowest between trial standard deviation and largest correct error trial differences. And this is a purely bottom-up approach. So we looked at data from 298 participants that completed a modified version of the Erickson-Flanker task. And we used multi-level models to examine a multiverse. So here I'm showing the number of decisions that went into each data processing stage. You know, in the grand scheme of things, only a few parameters went into each data processing stage, and there are many more we could have chosen. You could do an entire multiverse analysis just on different filtering procedures. So that's what I'm trying to emphasize. This is just a single multiverse. There are many other ways you could have looked at these data, and the code for analyzing it is all posted on OSF. Now let me show you the waveforms. On the top, we'll have error trials. On the bottom, we'll have correct trials. So if how we process the data makes no difference, we'd expect the waveforms to be entirely overlapping. But that doesn't seem to be the case for error trials or correct trials. There's quite a bit of variability there. But maybe it's just, waveforms are getting pushed up or down a little bit. So when we look at the difference between the two, they would be entirely overlapping. Again, that doesn't seem to be the case. So these are the difference waveforms, error minus correct for the ERN. And the peak of the ERN there ranges from about -4 to a little over -6, it looks like. There's a lot of variability in the scores we get depending on the pipeline we choose to analyze ERN. And all of these pipelines were based on parameters that are widely used in the literature. So here is a table only for completeness. I'm not going to go over it. I've been emphasizing this bottom-up approach. We are using this bottom-up approach using multi-level models to identify parameters to optimize for data quality or experimental effects. But we still need to use our heads. So you got to take this top-down approach as well. So what is ideal? So ideally, we could identify parameters that lead to high data quality and robust experimental effects, but only a single parameter had that. So only the region of interest approach for scoring ERN in this data set led to both high data quality and robust experimental effects. So low-pass filtering and the eye movement correction procedure didn't impact experimental effects at all. But it did impact data quality, so maybe that's reason enough to choose for those particular stages. Regarding measurement approach, so despite that the peak amplitude approach showed these nice, robust experimental effects, a peak amplitude approach likely misrepresents the true underlying ERP peak. So you might use a top-down justification that you might go ahead and use a time window mean amplitude anyway, and ignore the fact that a peak amplitude procedure led to the largest experimental effects. So this multiverse analysis approach of the data processing pipeline is just a way to optimize a pipeline for a particular purpose. And I hope it provides a roadmap for its application, and there's much more associated information on OSF and in the paper. But these different multiverses shed light on how different reasonable choices affect the outcomes of our studies. So in a preprint earlier this year, there was a tutorial advocating different multiverse analyses of psychophysiological data, looking at different paradigms for recording the same ERPs, different ways of process and analyze ERPs, like was done with this multiverse analysis, and then different statistical analysis procedures and how they can lead to different conclusions. Ideally, these multiverse analyses will show that these effects are very robust, but they're not fragile to any given small decision here or there in the data processing pipeline. Alternatively, maybe they will shed light on how fragile some of our findings are, leading to the identification of particular decisions that lead to optimal ways to look at an experimental effect, or showing that we might have just simply been capitalizing on noise for a bit too long. These multiverse analyses can help for studies that are trying to optimize ERPs for studying individual differences. I'm working on a paper right now with Greg Hychek And we're looking at the same multiverse analysis, optimizing ERPs for internal consistency across psychopathology groups. So I'm a clinical psychologist, and I'm interested in relationships between ERPs and psychiatric symptoms. And so these multiverse analyses can identify the parameters to help us identify the key processing stages to help us do that. And that's true for any study of individual differences, whether it's personality characteristics or educational variables, whatever you're interested in. And hopefully these studies and these examples and tutorials will help people apply these approaches to novel paradigms in ERPs, leading to more standardization in the field. And I'm not trying to suggest everyone should be doing the same thing, but I hope, very similar to the Many Pipelines project, that it'll give us a better understanding of how different decisions at any given stage can change a study's outcome. So there clearly is a need for improving the methods that we use. Authors, editors, and reviewers should all care about good reporting. And I don't know how much responsibility lies with each party for, the 2/3 processing parameters seem to be reported in any given study, but the way things are now don't seem to be working. So it's possible that these guidelines papers that have these checklists could become similar to PRISMA guidelines for meta-analyses. They just go hand in hand. These kind of guidelines checklists could end up in an appendix or posted on OSF. Pre-registrations and registered reports are helpful for identifying the relevant methodological parameters to consider in a study, because you're forced to think beforehand and spell out what are all the steps I'm going to take and plan to analyze how you're going to collect your data, how you're going to test your hypotheses. Another barrier to reporting might be that researchers don't know the specific ways in which the data were processed and analyzed due to reliance on various statistical software analysis packages. Because the appeal of prepackaged code is totally understandable, but code can quickly become a black box. To the extent to which popular software analysis packages can build in validity checks would be helpful so researchers can understand and judge whether the analysis approaches are appropriate for their data. And lastly, this sharing of pre-processing code and experimental paradigms will also help communicate what was done, because everything's laid out in the scripts. But I would not consider this a substitute for clear method section, because not everyone can be an expert in all of the different software packages and analysis pipelines. And even if it's popular today, doesn't mean we won't be using something else entirely different 20 years from now. So I hope that we have these clear recipes, that we understand the impact of any given decision or key ingredient of our stages, so that we end up with these beautiful, beautiful cookies. And I want to thank my co-authors, Michael Larson and Scott Baldwin. That QR code will take you to an OSF project for this talk, which will include my slides and references and other helpful resources. And I want to thank you very much for your attention.
Thank you, Peter. I'm not in the lecture hall, but I will give you applause. Wonderful. And internally, cutting EEGs want you for cooking EEG. So just let you know. So time for discussion locally. So we're going to turn off the camera again, 7 minutes, and then please vote or ask the question in the chats, and then we're going to throw the question to Peter in 7 minutes. Okay, so now we have a lot of interesting questions. Let's bring Peter back on stage, please. Can you hear me?
Mm-hmm.
Okay, a lot of things about cookie, but we put cookie alongside first. So first question to you. Can optimize the pipeline for effect magnitude lead to overfitting or false positives?
Absolutely. I mean, if you think about, the data processing pipeline suggested that these peak amplitude procedures maximize experimental effects, but those are likely going to lead to false positive effects simply due to the fact that you're mischaracterizing the true underlying ERP peak. So I would say yes.
Mm-hmm. Yep. And the next question is, how did you define data quality? Because you mentioned about that during your talk. And is that can be simple as an R, because some process or like a filter or some parameter that lead to a cleaner data and that also introduce distortion?
So we define data quality as between trial standard deviation of the score. I see no issue with using something like a signal to noise ratio or like Schimmel's RMSSD or even Steve Luck's SEM or standardized measurement error. Because the principle of a multiverse analysis is that you should only choose reasonable choices. So, I mean, I could filter my data so heavily that I end up with a flat signal and very high data quality, but it's going to obliterate any actual signal of interest. So as long as you're homing in on those reasonable choices, I think you should be safer than if you just kind of go willy-nilly and choose all possible filtering decisions, because in reality, many of those will not be used. And that'll also likely depend on your ERP component of interest. Like if you're doing air-related negativity, you're going to end up with a very different filtering than if you're doing something like a sensory P50 or auditory brain responses. So you still have to choose reasonable filters for your particular context.
In this case, another question, like because talk about the quality, and the one like audience curious, like you analyze reporting quality over 2 period of time, around 2012 and 2016. So did you see any improvements or change?
So that was the second motivation of the first paper, was to see whether these publication guidelines did anything, because the Keel et al. Paper was published in 2014. So we compared reporting and used 2 one-sided significance tests to basically show they're equivalent. Guidelines publications had no impact on reporting behavior on these 5 journals and articles published in them. So no improvement, no decrease.
So next question gonna be, is it valid to optimize for size of experiment effects without having a ground truth?
Well, I think by using the experimental effect, if it's robust and replicable, you're assuming that it's a real effect. And so that is kind of your ground truth. I mean, it's a difficult question, because if we had, say, real signals with no noise that represent some sort of ground truth, then we wouldn't be doing any of this anyway. So, I mean, it is a proxy for ground truth. It is a proxy for construct validity, for example, by saying we're optimizing for error minus correct trials differences. This should get at criterion validity of the signal of interest, that it truly is related to error detection. But I don't know how much for any of our effects we truly have some sort of ground truth. And we can have simulation studies.
Yeah. Similar question like many EG pipeline, right? I think we all kind of curious about how ground truth, but also have find a way to test it. The next question interesting from Cuba. So give a shout for CuttingGardens. Given that there is no perfect cookie in EEG, no ground truth about the thing we are looking for, is forcing a standard analysis the best way to proceed? Maybe better to follow different recipe to find different flavor and variants to fit our test?
Exactly. Yes. So I'm not advocating standardization. So I appreciate this question. So I'm not trying to say everyone should do something the same way, but by looking at how these different decisions impact our study outcomes, I hope it gives us a better appreciation of the important stages for our recipes. So like there's a proliferation of data processing pipelines that are standardized, that are ready to roll out for any particular purpose. But I think they're a bit premature for a lot of ERPs because we don't know the best way to look at them yet. And so the way we typically have done things is based on tradition. So, the way I typically did stuff was because that's how I was raised in my lab that I grew up in. Or that's how a favorite paper I did it, so I'm going to do it too, to do what they did. But this ignores the fact that there could be other ways to do it that are maybe better. So yes, we need to keep looking. And I think these multiverse analyses are a really helpful way to do so. I think there was a really kind of provocative paper in my mind by Arnaud Delorme a few years, or no, very recently, within the last year, who's basically saying EEG data is better left alone. I think that's the actual title of the paper. Something I didn't even consider was not filtering or not using eye movement correction procedure. And he basically showed that just first order high pass filter is all you need, and everything else just introduces more noise. Yeah. So I don't know. We got to keep looking.
We have to keep looking. That's why we have a lead station. We have a ton of, from many EG pipeline to multi-verses. So the next question going to be linked to variability you just mentioned. There seems to be much more variability across pipeline in the late and early EEG components. Why?
I would guess that this is just due to the later ERPs being more associated with kind of longer cognitive and affective processes and the early sensory components, which are more, I don't know, robust. They tend to be less latency variability than there is for cognitive processes that just involve more. That would be my kind of gut reaction or thought.
Interesting. Yeah, true. Interesting. And the next ones, because of time, how computationally heavy are these analyses? If anyone wants to apply, right? I think it's going to be a question they need to know.
So these analyses that we did for this particular project, we used multi-level modeling using LME4 and R. And it took the computer an hour and a half or 2 hours, but it got there. So, I mean, compared to how long it takes to process EEG data, it's a trivial amount of time, I think. I think the processing itself and getting all the different stages was much more time intensive.
And the last two questions are there his risk taker for analyze EEG data in attentional experiments for avoiding much loss of signal.
I don't know. I think that's something these multiverse analyses can help identify. You know, if you go by Arnaud Delorme, we shouldn't be doing too much because we lose signal. And, you know, these artifact rejection procedures might be leading to less statistical power that's going to hurt our capability to identify statistically significant effects. But I don't know of good heuristics, and I'm always leery of heuristics because that's usually how we process data and kind of how things are based off of, even though they tend to just be based on tradition.
Yeah. And this question, I think, because we talk about the open science, right? In the end, you're talking about moving forward method. I totally agree with you about presentations and make everything more clear, like you just mentioned, clear method. However, there's a question like data sharing, open products can be time consuming and not very rewarded. So what's your thought on that?
Absolutely. Yeah, unfortunately so. It is very time consuming. It is not rewarded. The bean counters of the world are not interested in open science, at least in some places where I am. Not many people care about open science, but I think there are selfish reasons to care about open science. So by embracing open science practices, I know it makes me more carefully write my code, and I have other people review my code. I'm commenting code more carefully. I'm making things more sustainable by freezing repositories, putting all the data in one place with all the scripts. And so at the very least, it's helping future Peter a few years from now, when I go back to look at these data again, to reanalyze them for some project, to understand what I did, when, and why. I also think it's very helpful. You know, I have a lot of graduate students and undergrads coming in and out of the lab. You know, someone stops doing a project, someone else picks it up, has no idea where anyone left it. And these kind of data management procedures, I think, can be helpful in that regard. But yes, I think you can't ignore that many of these open science practices, in terms of being externally rewarded, aren't quite there yet, unfortunately. So oftentimes we create positions where we have to be amazing at everything in order to still be successful.
I agree with you. I feel that the future me will thank me, and I hope students see the way I did it, they will do it. So hope for the best. Okay, last question. This is really probably not related to the question, but really popular is, can you share your cookie recipe on OSF?
Absolutely. I have a delicious chocolate chip cookie recipes. It's amazing.
I hope we don't have many cookies pipeline problems. Like everybody in the end have a different shipment flavor.
It sounds like a far more fun project. I'm all for it.
Okay, that's good. So I hope I hope nobody gonna bug you about the repositories. Be careful what you promise for. This is recorded, this one. Okay, thanks, Peter, for your wonderful talks. I'm gonna take a break. So we bring to a little bit break now. Thank you so much for staying with us. I just want to give a shout for Farmford Garden. You might wonder why it's crocodile behind me. Actually, this is a chair. How do I say it? Children caring room. Farmford Garden amazingly provide child care for the speakers. So if they bring the children, they do provide these services. How wonderful. I think all the hybrid conference should do that, promote diversity, equityness, and what's another one? Inclusiveness. So thank you so much for staying with us. We're going to be back. What time? It's 4:15. Yes, we have a 15 minutes break, and then we're going to see everybody on stage. So the last talk from Artemis project talk about reporting results. So see you guys soon. Keep tuning. Oh, hi everyone. So, okay, I'm gonna back to the station. I hope you already come up with your cafe and then some small break. So in the next talk, our last talk, we're gonna wrap it out the thing. We're going to have kind of dynamic Artemis projects that are gonna be presented by Angela. She's advocating for the clear and precise reporting in ERP research. And great, show the slide already. So Angela, we are excited to learn more about Artemis project with you. You want to take it away?
Okay. Can you hear me? Yufan, can you hear me?
Yes,
fine.
I can hear you.
Okay, great. Hello, everyone, and hello, everybody in the online audience. And thank you for having me here. So my topic here today is something that has a long title of the Agreed Reporting Template for EEG Methodology, International Standard, and then ERP edition, but we will see that it is, you will hopefully come to see it as a simple and a useful tool. And I will continue a little bit in the same direction that Peter started his last talk, and start with the fact that we all who have done any kind of EEG research know that it is incredibly complicated. It involves a lot of steps, a lot of decisions, and each of them opens a new path and a new path, leading to the well-known problem of the garden of forking paths. And we are sometimes not aware of how incredibly vast the garden of forking paths for EEGs is. And I like to give this example from a multiverse study that we have conducted that is not so unlike what Peter has been doing with ERN. And we varied only 2 to 4 different options for 7 different decisions. And we ended up by multiplying in each new step with new options and then multiplying until we got 864 different pipelines. And that is only a small subset of the true variability and the true size of the garden of forking paths in ERP research. Sorry, Katarina, I think this is... Okay. So it's working again. And here we have another example of that. It is a review of 132 different N400 papers. And it shows us that ERPs are the complexity of the garden of forking paths in theory is also reflected in the practice, because we have here the variability of people are really doing in actual ERP studies. And we see that there are some parts in which people agree and use relatively similar things, but then there are others in which they diverge quite a lot, even when you wouldn't expect them to diverge as much. So for example, in this paper, in these 132 papers, people use 13 different baseline periods to measure the M400 amplitude, and also 117 papers that had ANOVA to examine their ERPs. They had 99 different ways in which they combined channels in their ANOVAs. And as a result, ERP and other EEG studies are also very difficult to describe precisely because there are a lot of details that need to be captured. Some of them are maybe not as easy to express verbally, and they're not easy to recall at the time when you're writing a paper. So you have to remember to add all of these details into your publication. And Also, the journal space is very tight, very precious, so you cannot write a 200 pages report for your journal article, so you have to keep the space tight. And between the two, it is not uncommon that some things that are relevant get omitted or written, shortened, so that they end up being ambiguous. And here we have a different example from the same review of ERP studies. And now each bar that you see no longer shows how many different options there are for a given methodological decision, but instead you can see the green bar represents how many people reported, like how percentage of people who reported a given methodological decision clearly. Yellow is either ambiguous or inconsistent in different parts of the text, and red is means that information has been omitted. And you can see that some relevant pieces of information are missing quite often. For example, the order of operations cannot be deconstructed from papers with any degree of certainty in more than a half of all cases, and we do need that. So as Peter said, it might be tempting to think that it is not as important if we capture the general idea of a study for the main audience, but it is not really so. Transparent and thorough reports, or as Peter has called them, recipes, are important for several reasons. And one of them is being able to evaluate a study. Has everything been done properly? Maybe there is an important error that is hidden in these details that we are not aware of, and that influences the results if we want to evaluate another person's study. Also, if you want to conduct a replication, it is very helpful to have a thorough report, because then we are avoiding these discussions of whether these seemingly minor details in the procedure are the result why our results of replications and the original studies may differ or be the same. So it is important that replications are as close as possible to the original studies. And transparency is also very useful to people who are trying to build upon other people's research and who try to contact corresponding authors and get to these details. And as my Artemis teammate Susie nicely points out, it is especially important for people who are new to EEG, people who are in relatively new labs, and who are not beneficiaries of decades of informal knowledge that is passed down in labs that have longer traditions. So they rely on our papers to learn how we have done that, so that they can repeat it in their own labs. And finally, there is the meta-scientific perspective, that people who are trying to do systematic reviews or who are trying to do meta-analysis, they often complain that they have lacked information that they needed in order to conduct a meta-analysis. And there is one thing that I didn't mention, but Peter did, and this is the ethical aspect of the resources that we are wasting by not communicating everything and then having to go back and forth, and then perhaps wasting time in some directions that could have been avoided, and so on. No. Oh, I'm sorry. Please. And this has been recognized not for quite a while, but from the very start, because starting from the first guidelines on how to do ERP studies, which were published in 1977 by Donchin and colleagues, an important part of guidelines for good practice has been guidelines how to properly describe ERP papers, and they have grown over the years, and we have had also specialized ones. So here there are only a couple of examples of these guidelines. And in the more recent years, as ERP research has grown more and more complex, these guidelines started including checklists to help people remember all the details that they need to go through, because the list of what we need to describe is getting longer and longer. But unfortunately, this hasn't turned out to to help transparency in practice increase as much as we would hope for. And this is the study that Peter has talked about already, that compares the reporting practices before and after publication of the guidelines by Kiel et al., which included a checklist of the items that should be described. And we see that there is no increase, immediate increase in the next 3 years in the journals in which you would expect to be early adopters and among the first to enforce using tools such as this checklist. And we wondered whether there is something else that we might try to do to help in this case. So maybe guidelines are not the best possible way, and the direction in which we went was to think whether we could design a tool that would help people do their reporting in a more thorough way. If the problem is in not being able to fit everything in the journal, or if the problem is in not being able to remember everything on spot, or maybe reconstruct it several months after a study has been conducted. And this is the concept that we came up with. So we wanted to create a questionnaire that would include, so unlike a traditional checklist that would require you to just to go through it and check whether everything is in the paper, the Artemis form requires you to fill in information in a sort of a template that is standardized for every study. And the template is dynamic and branching, so depending on what decisions you have made, some of the questions show and the others do not, because otherwise it would be very, very large and overwhelming. And it has a user-friendly interface, so it looks like a regular form, like a Google form, to the end user, which produces on one hand a human reader-friendly output that can help somebody check, okay, I cannot find this information, is it there in the template? Or on the other hand, there is a machine-readable output which can be used by meta-scientists to aggregate and extract information more easily from a larger number of studies. And these are some features of this. So it is a questionnaire that is filled in, and it is intended to serve as a supplement to a publication. It is meant to document what has been done in a study. And it was built upon the experience that we have with systematic reviews, and also upon the experience of different fields such as aviation or surgery that have experience with checklists. So we have looked at what helped them make their checklists useful, and we looked into checklist literature in order to design our form in a better way. And in order for it to be truly useful, we designed it to be easy to fill in, to save time, because once filled in form can be copied. So you can create, for example, a template with a description of the hardware equipment in your laboratory. You never have to do it again, because you can just copy and then change the details or fill in the details that are missing. And in order to make it useful, we invited others from the community to contribute, and you will see that we were open to community feedback at different occasions, including this one now, to improve the tool that we have and include a diverse group of contributors to our design. It also allows documenting and saving information as it happens, so that you do not have to reconstruct it several months later at the stage of writing the paper, when it has maybe already gone from your long-term memory. And as I mentioned, it is easy to look at the details, but it also has a form that is useful for coding and for data, for information extraction on a larger scale. And the idea is that this tool can be used as a supplement to a publication with all other details, and then maybe you can feel a little bit more relaxed to squeeze in some parts of the method section because you know that it is available there, or you can use it to as a memory aid when writing the paper to make sure that everything is included, even if you don't want to write the supplement, or if you want to have information in both places. And it is intended to be used as a record that allows metadata extraction, because at the moment it is a very tedious process. And if anybody who has done a systematic review of literature knows how tiring it can sometimes be to, for example, look into degrees of freedom to check how many participants somebody really had, and what does one participant was excluded mean? That do you subtract them or do you not subtract them? And many other issues in interpreting and extracting information from verbal reports. On the other hand, it is important to note what Artemis isn't. And the first thing is that we often get feedback that it sounds very exhausting to fill in. Artemis for ERP has about 350 items at the moment. And that sounds like a lot, but because of the branching structure, you skip all the branches that are not relevant for you. And when I tested and I timed myself, it took 10 minutes to set up a laboratory hardware section. And it took me 45 minutes to describe a study from scratch, so without a template that saves time. And for example, it took me another 40-ish minutes for a multiverse study to, once I had one version, to just change and describe each pipeline in my multiverse section. So it is not that long when you think that it is a document that will stay as a record. It is also not intended to prescribe and limit what should be done, but to help you describe what has happened. So it is not a set of guidelines for good practice. It is not intended to say what should be done, but rather help you explain what has been done, and it is up to you to decide what is good and what isn't. And we once got an interesting feedback that says, I do not want to read the method section like a table to try to understand what has happened, and indeed it sounds terrible. So this is not intended to replace the method section and you can skip it completely, but it is there to help ensure that all methods information is there if somebody would want to look it up. And in the end, it is not a perfect tool yet. Probably it never will be, because ERPs are very complicated. And there are more and more details, like going down the rabbit hole when trying to build in new questions and ideas. But it still helps capture more information than is typically included in a paper. So even if it is not perfect at the moment, even if you don't fill in the whole template, it is not obligatory. There is no police that makes you fill in 100% of the report. But if you fill in 80%, that is still more than Peter's 63% that people report. So 20% more information is still okay. And this is what Artemis is in concept. And when we envisioned it, we wanted to go for all of the EEG, time frequency, EEG bands, ERPs. But we started from ERPs because that is the expertise that people that gathered in the team had the most. And also we had a systematic review of ERP literature that we could build upon and use as a starting point. And what you see over there is a network of the multiple choice questions in the ERP template and how it can lead you down different branches. And as I said, if it looks large and possibly overwhelming, it is not like that in practice. It looks a little bit different, and we'll see in a second. It has several sections, so study description, like title, DOI, and so on, experimental design, really basic features of it, hardware description, data acquisition, pre-processing, measurement of ERPs, selection of ERP channels also for measurement purpose, visualization, and additional comments. And it doesn't include statistics because this is a whole other field that will probably end up being as large as this one already. And one form in the current, so one document, one report, would describe one experiment with one component. And if you have a complex experiment, at the moment, the workaround is that you can have, for example, 2 or 3 Artemis reports for one single experiment that has multiple hypotheses. And this is what Artemis looks like if a person logs in. So it has an option to review your own templates, to browse through others' public templates, to create new ones, to upload ones that you have already saved on your computer. And I circled down there our laboratory template that we can now reuse and save time, and also help younger colleagues in the lab that may not exactly know what is our online filter settings or something else. They can look it up there and they don't have to brush the dust of the manual. And this is what the form looks like. So it is very much like a simple Google Forms thing. It is just not as simple in the background because it has a lot of options, a lot of branching that happens in the background. And how am I going with timing?
Almost there. I think maybe 1 more minute.
Okay, so then maybe let's not go into the details of how this came about. I will thank the team in the end, but I also wanted to thank the Cutting EEG group who has helped us launch this project a lot, because it started, our kickoff was at Live MEG in 2020, so I'll just use this opportunity to say that and keep the rest of the story for another time. And well, there are also some challenges in doing this kind of research that are shared across similar projects that are focused on building tools rather than conducting studies in ERPs, such as finding time and people and funding agencies that will be willing to fund this type of work and not classical research. And then ensuring version compatibility when new versions come out, engaging community, and so on. But perhaps we can leave that for discussion time, because I would like to use this opportunity to invite you to, in case you are interested, look up the Artemis statement that tries to spell out the basic idea behind Artemis on one hand. And I would also like to invite you to an open doors Artemis meeting that happens in our Gather Town space, which you can see in the background, and it will be on 17th of October. And the time, the hours will depend on your interest, because people are listening from different time zones. And this here is a Google form, the QR code, and I can also share it if you would want to on the crowdcast later. But if you are interested in joining, there is a Google form, and we can set up the time that would be convenient for people depending on the time zone from which we have interest, because probably Singapore and Los Angeles wouldn't have a lot of hours in common. And the open door session will be intended for people who are interested to learn more, but also to contribute possibly to future developments of art. And that would be it, I think, from me. So I hope I'm not over the time.
No, you're perfect. That's good. So maybe leave a slide if people want to scan the QR code. Anybody's interested in joining the project?
Sorry, 17th November. Okay, I see that the audience is looking at me in an extremely confused look, and 17th of October was yesterday. So
it's November, right? Because October yesterday. We cannot join.
Exactly.
Very exclusive. All right, so thanks a lot. So we're gonna turn off the camera. So thanks a lot for great talks. So there are some questions for this project. The first one, how do you recommend referring to the Artemis form of study, linked to Artemis website or supplementary? I think this question, maybe they want to know how do they references this project, like website or study.
So we have a preprint that we hope to submit somewhere soon, and we can share the link to the preprint in the comments. And that would probably be the best way to cite it at the moment, because we don't have a paper that has... We have a concept paper, but for the Artemis for ERP, it would probably be the best to cite the preprint describing the tool itself, not the generality. And we can share the preprint in the link.
Okay, so one question from Cuba. Does Artemis already have a shorter or specialized version for clinical report?
No, it is a general form, and it should be the same because it is generic, so it should be the same for clinical or developmental or any kind of other kind of research. But if there are some sections that you want to skip, if you want to have a shorter report, it is not obligatory to fill it in, although the report highlights which fields have not been filled in, so you can easily see what has been skipped. But you're not required to necessarily fill it in.
Mm-hmm. So the next question is more about, because you mentioned about the method and how to fill out the form. And the question is, will it be possible to automatically generate a method station test based on a field Artemis form, the web app?
That is currently in the domain of science fiction, I guess. But maybe someday with Artemis and ChatGPT on a shared goal. I don't know. No, at the moment, no. In short.
Mm-hmm. Yeah, I think also generally, we cannot write exactly the same method station. This is the thing I have. We're kind of similar in terms of reporting the result, but we cannot write exactly the same, right? Okay, the next question: do open science standards according to which experiment code and analysis scripts are shared make reporting standard obsolete? Sorry, the questions need to digest a little bit.
Which one do open standards according to? I wouldn't say so, because at least in my case, if you share code and share analysis script, not everybody is equally familiar with the program that you use or the language that you use. So even though you share, and it is not always equally accessible, and it is much more human readable to have a method description than to have to dive into somebody's code to try to understand what has exactly happened. So I wouldn't, as a reader, I wouldn't like to have to read through the code in order to understand what has happened. Although I would like to have the code in case something is not clear, so that I can check it, or if in case I want to replicate the same procedure in my lab, then it would be nice to have the code as well.
True. And next questions, we talk about flexibility in this whole session about in terms of analytical pipeline. So how flexible is Artemis, this web app or the project to extensions, since currently it was focused on ERPs, right? For example, IEG.
So it is relatively easily expandable into other subdomains. ERP is just the first, and it is modular, so experimental design can be shared with the different subtype, hardware can be shared, acquisition can be shared, so different subsections of the form can be reused for a different kind of research and then built upon. We just haven't got there yet, and it would be very nice to have people who are experts in specific subdomains who would be interested to work on something. For example, I have never worked with IEG, so I wouldn't feel comfortable in doing something like that on my own. We need a domain expert.
Mm-hmm. Yeah, similar. I think you answered already another question about whether it's suitable for other types of study, right? Not ERP. So next question is about the decision, because we all know we have to choose a parameter in terms of report. And what happens if analysis doesn't fit into the decision tree, like the one we use the web app? It doesn't fit into the structure. How to report using the Artemis web app?
So that shouldn't happen too often because we covered most of the basic designs. But in case you do have something very specific that is not there, at the moment, what we have as an option is that we have an additional comments field where a person can describe any extra things that do not fit anywhere else. And for example, I added information about different thresholds for EOG and EEG that were used in a study. And so I just put it in an additional comment section. It's a minor thing, but it can also be used for more complex things. It is not ideal for meta-analysis, but it is still easier than working with a journal article.
Mm-hmm. And next question is a kind of general comment. I think it's interesting since we don't really have so-called standardized pipeline, right? So someone's a last like beginner or not familiar with that. In this amount of standardized pipeline we were able to choose, do you have any advice?
In the ones that we can choose in the Artemis template, or
yeah, I think from the template you do have a lot of parameters, right? Like you have steps through a well document. Maybe perhaps it's good for beginner to have idea what kind of pipeline will look like.
Well, Artemis is not prescriptive, so I'm not an expert in standardized pipelines, so I couldn't tell anything about choosing one versus the other. But I think the amount of standardized pipelines that we have tells us something about this field. So many different solutions, each of them wanting to be more general. So it is challenging, but it is just we offer what is there in the form, and you can choose among them the one that you think is better, or you should consult somebody who is an expert for that. So Artemis is only there to describe and not to advise.
And you mentioned about this web app. In the end, you have a JSON file like an output. So it's kind of used when we were going to submit the paper, let's say. So is there any initial experience as to author who use this app, the Artemis web app, for their manuscripts, and how editor and reviewer responded to it, especially when used to abbreviations, the traditional methodization?
That is a very interesting question, but we don't have any experience at the moment. I'm about to add Artemis reports into one paper that I'm submitting for revision, so I will see how they respond as a first personal experience, because it is the form that we have in this current state is relatively new, so we don't have any papers coming out yet that have used Artemis. Yeah, but we will see. Hopefully they react in a positive way.
Yeah, I'm also curious how they receive already standardized output. So we talk about standardized pipeline. The last question will be, is Artemis compatible with bits?
At the moment, we have tried hard to use terminology that is consistent with bits to make sure to know which fields in Artemis are also available in the bits format, but we are not translatable in any way. So you cannot import from a data set that is formatted with bits and just get automatically information in Artemis, or in the opposite direction. And also, I would say that each of them has some things that are shared, and also something that the other one doesn't have. For example, and for those who are not as familiar with bits, so it is a standard on how to format and structure files, folders, which information should be included in a data set. And it is very detailed in some aspects in which Artemis isn't. For example, it offers you to describe each electrode position in the 3D space and how they translate into channels, and many, many technical details that would be just too much for Artemis. So Artemis asks questions, the same ones that reviewers should expect you to already include in your paper, not more than that. And on the other hand, BITS doesn't have information on rationale. Why has somebody chosen a specific thing? Why have you used this time window to study your component, or this set of electrodes? So the decision-making process is very important, but it is not captured in the data itself, and Artemis includes it. So they are kind of like two on a Venn diagram, they would overlap partially.
Thank you so much. It's really wonderful talks. So let's bring to our final discussions. So first of all, I want to thank everyone's attention staying with us, and thanks those three wonderful speakers show us your projects. I also would like to highlight Managed Pipeline Artemis project, really led by a lot of ECRs who really do this job voluntarily. Let's address this hard question, right? We can see how many garden or forking parts in the ERP research, and either more, can we apply to clinical, can we apply to different field? So I assume maybe a lot of you have a lot of questions about those kind of projects or the result analysis, etc. So the following, I'm going to give 5 minutes. We're going to maybe turn off the camera like we did, and then we follow up. We have the question for our speakers. From here, the catering donate 100%, not 10% of profits for the social purpose here in the Garden Frankfurt. I feel really amazing about that. So welcome all the speakers. Thank you so much, Elena, Peter, and Angela. So I would like to start maybe the first question, something interesting, maybe from the first outvote first then. So we can paste on. I think maybe the first question we have wrong from everyone. First one is quite long. Let me read it out. When we speak about different forking paths, we assume that all decisions bring to the different destination results. But do we have evidence that all forking paths lead to clear different results? For more concrete example, do we have evidence that reference mostoid given a clear different result compared references maybe on average for a specific ERP? Or change are marginal? I think this question can have of like each speaker maybe can share your thought on that. Who want to start first? Peter, would you like to start first?
Peter is muted.
Oh, and Angela or Elena, which of you would like to start first?
I can go.
Okay.
Or Elena, either way.
Okay, Angela, I think you are ready.
We do have that kind of evidence, and I mentioned that multiverse study, for example, that's something that we did in our lab that I'm aware of, but there are other similar studies. So we did a multiverse analysis in which we compared in each step how different decisions affect outcomes in terms of data quality, statistical power effects. And we see that they can affect results. Some of them can, especially the ones that are related to statistical analysis, can influence greatly results, even the big effects. And some of them, such as filtering, can affect only very small between condition differences that you may be interested in, because usually it is not your goal to show that, for example, a P300 exists or an N400 exists. We already know that. We have known for 40 years, but you're interested in some smaller difference between conditions, and those are more sensitive to variations of this kind. So, and depending on the component and the feature, so we can get into more details, but maybe that would be too much.
Yeah. Thank you. Elena?
We didn't do those analyses yet, so we can only suspect what will have a bigger or a smaller effect on the results that we're seeing. So I'd rely on two other speakers a little bit more. I suppose they do much more of the multiverse analyses, or did them up to this point.
And Peter?
I would say there's already evidence. Can you hear me now?
Yes.
Wonderful. I would say there's already evidence that these kind of different decision points can impact the study's outcomes. So, for example, Anna Weinberg's group in a paper led by Iceland Sondre, they showed that different data processing pipelines led to different observations of clinical group differences between controls and anxiety people, or even gender differences in their ERPs. And the preprint I mentioned in my talk, it does a multiverse analysis of the ERN anxiety relationship, showing how these different choices that you make lead to different conclusions on whether ERN is related to anxiety and how you analyze it, or the different paradigm you use for recording, or the different sensors you use for analysis. So I think they can and do, hopefully, sometimes they don't, but right now we're just kind of guessing.
Mm-hmm. Yeah, and I'll lead to another question, the next one. So we are talking about all the flexibility and different choices, how that affect our result, right? So do you think that it's a plan to develop a standardized pipeline for statistics analysis? I saw that a lot during all of you guys talk. People ask as well for statistics analysis and reporting as well as pre-processing. As I think it's the same question for all speakers. And feel free to start with. Maybe Angela? Yeah.
So I think there is already multiverse analysis approach, and in the initial study where the concept was piloted, if I remember correctly, it was applied to statistical analysis and not to pre-processing of any kind of data. And in the N400 paper that I mentioned, we compare measurement and analysis choices as well. But I guess in that sense, the EEG many pipelines will be able to compare many more statistical analysis choices, because they have a big sample of these different approaches.
Mm-hmm.
But I guess Elena can say more on that.
Elena?
So we are using the statistical analysis choices that were provided by the analyst teams. So we are exploring only the ones that exist there in the practices. We're not exploring all of the possible choices. That would be a bit of a different multiverse analysis approach that other groups are applying. And as far as I know, even if you take into account the opinion of established researchers to recreate all of these pipelines, it could result into thousands of different choices. We'll see. I think it's a very good question. Part of it, if we're aiming to standardize practices, I think we are really aiming to understand them and map them, because we don't know that up to this point either. And I think certain level of flexibility is definitely necessary to keep there. We cannot standardize all of the practices, but we have to understand which steps affects the data and how does it affect it. Mm-hmm. And we have to communicate it further to know. Maybe just to say shortly, as an outcome of the script check that we did, one of the side things that we saw, and what was very interesting for me to see, is to have the list of functions that analysts have used and how many teams actually used all of these functions and what does it actually do. I think we don't even have such a source, and it will be beneficial because there's also a garden of working parts of all the options that are possible there out in the world, open source, that you can take and use, but it's not so commonly known how often these practices are used in general. And that's like another thing I think is important to consider that as well.
Yeah, sure. Sorry about the confusion. The question still stay with the last one. I think we're going to update the question. This is going to be the next one. But still, like for Peter, do you want me to repeat the question, or you?
Oh, I remember it.
Okay, cool. Thanks.
So yes, there already are multiverse analyses of the statistical analyses that are used. So in that same preprint I mentioned, we did a tutorial on how to do these for psychophysiological studies. And so what we did is we basically looked at plots of p-values from all the different statistical analysis approaches we did. It's a bit harder to compare than doing something like a multi-level model on the same data processing parameters, because it's a little more subjective looking at these plots of p-values or effect sizes. But as was mentioned, that was how these multiverse analyses kind of originally came about in that first Stegen paper.
Mm-hmm. True. So the next question you probably see on the monitor is very interesting about is any deep trends in data analysis come from? Are they coming from tutorial for software package? We know there are some paper talk about that. Or what about the default value of parameters? Sorry, it's an echo. In software package, does anyone have any experience or like thought on that when you look into your data or look into your project? Elena?
Can you hear me now? We were also checking where this echo is coming from. I'm not sure if everyone hears it. Yeah, that's a good question. I think it was partly also that for EG many pipelines. And I think there are many possible ways to think about where these practices come from. So one of the possibility are the tutorials that people read online. However, I think that many codes are shared between the teams. So it might be really that it's coming from one single hub. And I do believe that we don't always know why we do things in certain steps. I guess it's because that's how we learned and that's how we continue. It's also, since I observed this variability between the software packages, I did start asking around people that I knew, for example, are using MiniPython. I would ask around, why would you do it in this particular step? And they all have really good answers. It all completely makes sense. So I think we just have to foster the communication between different teams, like doing pre-processing data in different ways, just to open the communication and start hearing each other. What are the reasons for doing so? With the tutorials, I do believe they also have a huge role to play. I think once we take a package or analyze our data in a way we follow the tutorial, and it has a high impact on that as well. However, I was also advocating for keeping your eye always on it, so never using it blindly and exploring the data. I think that's an important part as well, but it could explain partly the variability that we see across different softwares.
Okay, Peter and Angela, do you want to add anything for this question?
I think the only thing I would add is I have no idea, but I think these tutorial papers are very influential in this regard with their default values. And a lot of times there are very justified reasons for why people do the things they do, but sometimes that can get lost. And so, I think writing your own code, for example, to do your own filtering to your data, like shows you why you tend to do filtering on continuous data instead of on segmented data. But I don't know. I don't know where these trends come from. But hopefully it's because principled people already thought about this 40 years ago, or I guess almost up to the 100-year anniversary.
Yeah,
even though the impact
that's true.
until we look. Yeah? Sometimes we're like reinventing the wheel when people already wrote about this 30 or 40 years ago, but we failed to read the old work.
Mm-hmm. It's true. Angela, do you have any?
Because people usually don't describe that in their papers, we don't have objective evidence. And I can imagine defaults in software being important, especially if somebody doesn't have many other sources of information to look for, and then they go and trust the designer of the software. At least I've seen that it happen. And when it comes to anecdotal evidence, what a lot of people that I know do is that they ask somebody that they trust, they go to somebody's lab to get trained, they read the paper, and then they say, so if I want to dive into a new field of research, let's see what other people have done within this particular field. So there is a lot of seeing what other people have done and then doing the same thing, just to have a base to start on. So but that is only anecdotal evidence, and only to add as another idea what might be driving trends, but there might be many other valid reasons that I wouldn't know of.
True. I wonder if any audience would like to on stage. If you have any question you would like to ask and can think about that, we can prepare you on stage. You can tell us in the chat. Just let you know, you can get prepared. So I'm going to ask the question. So if you have any question you would like to ask those speakers, please do so. We're going to close this session 5:30 sharp. So feel free to ask any question at this time. So next question, I think is interesting. We talk about open science, and then we do those, we talked about before, all the kind of make everything transparent and document everything, data time. And another question can ask, like, can this idea of multiverse analysis help to promote slow science? Peter, what do you think? Yeah, I don't know. What do you guys think?
Honestly, I would hope so, because if I don't know, maybe it is our lab and we are slow, but everything that we wanted to do thoroughly and well always takes time, and things that get like, oh, let's do it in a shorter period of time, then you have to make some compromises in order to respect the tempo. So I wouldn't mind, but maybe that's just me. I like working slow, but in a way I'm comfortable with. That might be personal preference.
So other people think, do you think that help to promote slow science? As long we all know the pace is quite fast, right?
Yeah, I'm not sure I know what slow science is, but in terms of incremental science, kind of like Margaret Bradley talks about the scientific pendulum swinging back to incremental science, we start only, focusing in on one piece of a study and changing it to see what happens. I hope these kind of multiverse analyses promote this kind of incremental gain in knowledge, so that we don't make these big leaps and not have any idea how we ended up there.
And make sure the results are replicable, right? That's what we want to do now. Elena?
I agree with Peter. I think it does promote slow science, but in a possibly the best way possible. I think it increases the robustness of science, or it has the potential to increase it. So it's just to say that if you have one pipeline or analysis choice that you do, you got the result, you're ready on to go and publish it. But the multiverse analysis will stop you there, and you're like, wait, let's check how robust this result is. Let's try to run tens or I don't know how many studies, pipelines on it to see really what's the variability in the result. And that can really give a very good estimate on how reliable or how stable a real result is. And it's definitely, I think, beneficial for us to know that as well.
Yeah. And next question is kind of interesting, and about those, when we talk about open science, those beating science projects. Many of these projects are not funded and are run by researcher personal incentive. Is the lack of funding a problem?
It is. I had the chance recently to take part in a meeting of several open science projects that work under the umbrella of the INCF, which is the International Neuroinformatics Coordinating Facility, which is this very nice organization that supports projects such as Artemis, but also different kinds of standards within the field of neuroscience and neuroinformatics. And pretty much all projects had the same complaint, that these kinds of things are a side project that we work on when we have time, when we can afford it, that there are no calls. So there has been an agreement, and it's not like a personal impression of Artemis, but something that has been echoed around, and people have been lobbying to the INCF to lobby to other organizations to do something about it. But who knows? Maybe somebody in the audience can do something about it and hears this now.
And other people? How about Elena? What do you think for mini each pipeline?
I think we recently had this discussion a little bit as well. I do think that funding is important for these projects, especially when they're dealing with a lot of data and a lot of computational power. In our project as well, I think the community support is also equally important. So these things, I think, are two of the most important things as well. So funding, I think, is not the only important thing there, but it's definitely important when you're dealing with a lot of data and you have to have, time, tools, equipment, clusters to run all of these analyses. I mean, it's really hard to go around it or without it. What's also important is the support. By your peers, by your supervisor, wherever you're working at. yeah, I don't know if I have much more to. To my personal opinion is that I think we should fund more of these projects. Honestly, I think they're extremely important for the future of science, and sometimes we do not give enough of credit for that.
But I think more and more we got awareness. Yeah, Peter, sorry.
Well, I was just going to say, I think it's hard to know because funding varies so much by where we live and work. But I think part of this might more be on us than on the funders, and that we haven't convinced them that this type of work is worth funding and showing how monumental these findings can be and promote active science. Like so I don't know. A lack of funding is always a problem, no matter what field you find yourself in. But it's up to us to convince the funders that this work is worth funding.
Mm-hmm. Okay, true. Okay. Last question from Cuba. Do you think resting state EEG experiments will be more difficult to reproduce, like in terms of like clinical setting, than ERP studies? So will it be possible or worth it to look for standard reporting and pre-processing in this content? If anyone have the experience, please share your experience. I think Peter, right? Because you mentioned you have some, or maybe I misunderstood. None?
Sorry, I know I haven't done resting state, although EEG Many Labs is doing a resting state replication project. So maybe they'll answer this question for us in a year or two.
Elena, yeah.
I did quite some resting state EEG for my PhD work. It was not reproducible science, but I think I could have a gut feeling that the variability of the results we see in EEG Many pipelines for the time frequency domain would be quite similar or overlapping with the resting state work, because you mostly look at oscillations there and compared. However, if you are asking about the clinical setting, if we're taking patient groups or similar different groups, I think it might. I mean, my suspicion would be that this will also influence the variability in the data as well, because the data as default is quite more difficult to deal with, to clean it. You have, there's a lot to think about. So I think it's generally generalizable. If you're more interested to it, I would guess that time frequency analysis would give a rough estimate. Maybe that's the closest that we have to look into it. And the clinical samples could be something that, again, one would have to look into in more detail. There could be some deviation for that reason.
Thank you, Elena. If nothing from the audience, is any audience would like to have your final shot of the questions? No. So thank you so much for the three speakers. And this, I really enjoyed it fully, as I'm really involved with those two projects. And in the beginning, we're talking about the variance kind of analytical pipeline to multiverse analysis, and then down to how we can make it better by reporting the result in a more transparent way. I do think this is really nice talks by three speakers. And thank you everyone from tuning. And today I need to thanks I want to bring my team. Apart from Crocodile, we have Nat. We have two Natalie and Nanis that help us to choose the question, and so without them, I cannot manage to filter all the questions. So given a lot of posts, and also in the lecture hall with our Felix as well there in our lecture hall. So thank you all for tuning, and thanks everyone for joining from all over the different garden and work, and thank you so much from different time zones as well. So wish you have a nice evening. We're going heading for our social event. Don't get jealous. So so I will see you, some of you here in the Frankfurt Garden, and I wish you guys have a nice evening. And tomorrow, bye.
Thanks, bye.
Thank you, bye.